Good evening, everyone. Now, in my previous video, I had started up with the enzyme inhibition and I finished up with allosteric inhibition. The reason why I took allosteric inhibition because it is very important from examination point of view. And I mentioned in that video that rest of the different type of inhibition I will be discussing in my next video. So here I will be discussing now the rest of the different type of inhibition. So let's start up with the discussion. Now, I told you what are the different type of enzyme inhibition. This chart I showed you in previous video also. So we had discussed this aspect, allosteric. Today we are going to discuss the reversible, which is of three types: competitive, non-competitive, uncompetitive. And irreversible example is suicide inhibition. Now see, enzyme is such a topic which is actually important for every competitive exam. Whether you are preparing for your MSc entrance biochemistry, whether you are preparing for your NEET PG exam, whether you are preparing for your FMGE exam. Whenever we talk about biochemistry, the concept of enzyme is bound to come. Right. Now let's take up, first of all, you should understand by the term reversible, as it is clear, which can be reversed back, or inhibitor bind non-covalently with the enzyme. So if there is a non-covalent binding, automatically inhibitor is removed. So action of enzyme will be fully restored. An equilibrium is established between the free inhibitor and enzyme complex and is defined by an equilibrium constant. And the activity of the enzyme, main thing is the activity of the enzyme can be fully restored. If we increase the concentration of the substrate also, then also the reaction can be reversed back. So first of all, we are going to discuss competitive inhibition. Now see, as the word indicates, there is a competition between the inhibitor and the substrate. Means inhibitor resembles the substrate in structure. So it can be reversed by increasing the concentration of substrate, isn't it? If I show you the reaction also, you can see here, this is the enzyme. This is the inhibitor which is resembling, and if the inhibitor combines, then no desired product will be there. And if the product combines, automatically, or you can say if the substrate combines, the product will be produced. Right? Or in case if we have to explain competitive inhibition in terms of Vmax and Km, Km increases in competitive inhibition because the affinity of the enzyme will substrate will decrease, and if we talk about Vmax, it remains the same. Right? This is a very good depiction. You can see if the substrate is combining, simple enzyme substrate complex, and product is produced. But if the inhibitor is combining, no reaction. So, or in other words, if I have to write it like this, enzyme will combine with the substrate, forming enzyme substrate complex, which dissociates to form enzyme and the product. And if inhibitor combines, it forms enzyme inhibitor complex. Right? And this is Lineweaver-Burkes plot. This is very, very important. Questions have been asked on this. Lineweaver-Burkes plot, which is actually a reciprocal of Michaelis-Menten equation. Okay. And if we look at this, you can see basically Lineweaver-Burkes plot explains us the concept of straight line. We can calculate the slope, we can calculate the intercept. So यहाँ पे you can see no inhibitor, inhibitor. So what will happen? There will be a Vmax will be the same. On the other hand, if we see Km, just now I told you Km will increase. So Vmax is not changed, or Vmax is same, whereas Km is increased because the affinity of the enzyme towards substrate is decreased in the presence of this inhibitor. This is very important, and the questions are generally asked on this aspect also, right? And for example, malonate is a competitive inhibitor of succinate dehydrogenase. Succinate dehydrogenase, which is an enzyme of TCA cycle. So basically what happens is there is a structural resemblance between malonate and succinate. So if succinate dehydrogenase takes malonate, no fumarate will be produced. Is it clear? Then another therapeutic use, antibacterial action of sulfa drugs, which are the structural analog of PAPA, para-amino benzoic acid. Right. So इसमें भी again you can see there is structural resemblance, so this synthetase won't work. So sulfonamides क्या करेंगे? They will inhibit the bacterial enzyme. However, they are non-toxic to human because they cannot synthesize this folic acid. So this is another very important example of competitive inhibition. Then there is a therapeutic use of competitive inhibition. Very, very important. The questions are generally asked on this. Now, if we talk about statins, which are basically anti-hyperlipidemic drugs, or which actually decreases the concentration of cholesterol given to the person suffering from hypercholesterolemia. So the enzyme there is HMG-CoA reductase, which is actually the rate limiting enzyme of cholesterol biosynthesis. Right. Then comes allopurinol. The enzyme is xanthine oxidase, and this allopurinol is given to cure gout. It is given to the person suffering from arthritis or increased concentration of uric acid. Then methotrexate, which is an anti-cancerous drug. It is an inhibitor of dihydrofolate reductase. Caprotryl and enalapril, angiotensin converting enzyme, which are actually given to the person who are hypertensive. Then dicumarol. Dicumarol is actually an inhibitor of epoxide reductase, which is actually an intermediate produced during vitamin function of this vitamin K. So these are what? These are the therapeutic uses of competitive inhibitors. Examination point of view, say they are very, very important. Then non-competitive. Non-competitive means no competition between the inhibitor and the substrate. Or you can say inhibitor has no structural resemblance to the substrate. An increase in the substrate concentration does not relieve this I, because I and S, inhibitor and substrate, are binding at different sites. So if you look at the reaction, if we see Vmax will be slowed but not halted. And I'll show you the equation also. You can see substrate is binding here, inhibitor is binding here. So automatically inhibitor can bind with the enzyme and inhibitor can bind with the enzyme substrate complex also. So what happens to this is Vmax is lowered, slowed, but Km is unchanged. There won't be any change in Km. Right? Then we come over to the Lineweaver-Burkes plot. As you can see, just now I told you that there will be a decrease in Vmax. Right? But Km is the same. This is actually Lineweaver-Burkes plot. Km is unchanged, whereas there is a decrease in Vmax. This is what? This is non-competitive inhibition. And these are the different examples of non-competitive inhibition. Heavy metals bind with the cysteine group of E in the enzyme. Pepstatin binds with pepsin, soybean trypsin inhibitor for trypsin, and ethanol or narcotic drugs for acid phosphatase. So this is what is non-competitive inhibition, where there is no competition between the inhibitor and the substrate. Then we come over to the last inhibition, which is actually very rare, uncompetitive inhibition. Always remember the basic difference between different type is, you can see the inhibitor binds with only enzyme substrate complex. This is the major difference between these. And the example is, it is very rare, inhibition of placental alkaline phosphatase by phenylalanine. So this is uncompetitive inhibition. And if we see, you can say, if we talk about Vmax, it also decreases, Km, it also decreases. Right? So there is a difference between competitive, uncompetitive and non-competitive inhibition, leading to changes in Vmax and Km, but all will come under reversible type of inhibition. Then irreversible. Now irreversible, as the name suggests, it cannot be reversed back. Means enzyme binds covalently. Covalent bond is considered to be the strongest bond. When it binds with that, it cannot dissociate from the enzyme. So what happens is, there is a change in the conformation at the active site of enzyme, destroying their capacity to function as catalysts. Right? Just for example, number of variety of poison. They say iodoacetate is there, OP poisoning is there, oxidizing agents acts as irreversible inhibitors. The most common irreversible inhibitor is cyanide, which is an inhibitor of cytochrome A1, A3 oxidase. Now, under irreversible inhibition, I gave you a hint of suicide inhibitors. चलो, if it in terms of kinetics अगर हम देखें, तो irreversible is little bit similar to non-competitive inhibition. Means Vmax will decrease, but there won't be any change in K. These are the different inhibitors. As you can see, this is a very good fluoride, is an inhibitor of enzyme enolase. This is the question which is generally asked in the exam, because it inhibits glycolysis. Malathion, acetylcholine esterase, diisopropyl fluorophosphate, it is also known as nerve gas, and BAL. Right? Then we come over to suicide inhibition. Suicide inhibition is a specialized form of irreversible inhibition. So it is basically known as mechanism based inactivation, because inhibitor is converted into a more effective inhibitor with the help of the enzyme to be inhibited. So enzyme is actually committing suicide. That is what is written here. Inhibitor is converted to a more effective inhibitor with the help of the enzyme to be inhibited. So enzyme literally commits suicide. They utilize normal enzyme reaction mechanism and inactivate the enzyme. So this is what, this is suicide inhibition, a part of irreversible inhibition. Right? So I guess, and these are some of the examples. You can see, they say allopurinol. Allopurinol will come under, you can say competitive inhibition also, and it will come under suicide inhibition also. Similarly, 5-fluorouracil, this is a structural analog. Right? Aspirin, it is a suicidal inhibitor of cyclooxygenase, जिसको हम COX बोलते हैं, which is utilized for the synthesis of PGs in our body, prostaglandins in our body. And similarly, DFMO, which is a inhibitor of ornithine decarboxylase. So this is all about inhibition. This is just a quick revision of inhibition, and sure and short questions have been asked in inhibition in the previous years. So I hope the topic of inhibition in enzymes is clear to all of you. Baki rest, if you people have any queries, I have always told you to apply on my website, that is Biochemistry Simplified. Biochemistrysimplified.com. So basically my main motto is to make biochemistry easy for you guys, because this is such a subject which is asked in most of the exams. So I hope I would be giving maximum outputs, beneficial outputs to you guys in future. So next topic which I would be discussing would be, in enzymes only, the kinetics of enzyme, which is again a very important topics. Kinetics of enzyme, very, very important topic, because whatever topics I am taking, I am basically emphasizing on the type of questions which have been asked in the exam. So watch my video carefully, and always I have told you, keep a copy and pen so that whatever you find important, that you can just note it down. Because this is my personal experience. When you note out something, with your pen, automatically it goes in your brain. Is it clear to all of you? So I think this is more than enough for today. So in my next video, I will be taking up kinetics with you guys. Thank you so much. Take care.